Good afternoon, everyone. I'm Viktoria. My name is Viktoria Csajbok, and I came from a company called Femtonics, which is mainly known about its two-photon fluorescence microscopy solutions. But today I will present our other work, which is about intelligent image analysis in multimodal SRS microscopy. To begin, I would like to place SRS microscopy into a broader context of light-matter interaction. So when light interacts with a material, several different optical processes can occur, depending on the energy structure of the material and on the type of excitation. Absorption and fluorescence are mainly related to electronic transitions, while infrared absorption and Raman scattering probe molecular vibrational states. And this is important because the vibrational contrast gives us information, gives us chemical information about the sample itself. And this is the basis of label-free molecular imaging. Raman scattering is a nonlinear, is an inelastic scattering process where the scattered photon has a different energy than the incoming photon. And this energy difference corresponds to the molecular vibration. And it's plotted as the function of the Raman shift. And this Raman shift, the signal, is important because it's determined only by the molecule and not by the excitation wavelengths. This is the reason why sometimes Raman scattering is called a molecular fingerprinting technique also. In biological samples, the Raman spectrum can be divided into three main regions. The first one is the fingerprint region. It contains many molecule-specific bonds. The second is the cell-silent region. Endogenous biological molecules gives a weak signal in this region. That's why it's suitable for Raman tagging. And the third big region is the C-age stretching region. It's mainly dominated by lipid and protein-related signals. Well, classical Raman spectroscopy is very informative, but gives only a weak signal, and the imaging is very slow. And stimulated Raman scattering is the stimulated version of the classical Raman scattering. And here we use two synchronized laser beams. They are called the pump and the Stokes beams. And when the energy difference between the two beams matches the vibration of the molecule, then energy is transferred between the beams. So instead of detecting weak scattered photons, we only need to detect a small pump loss or Stokes gain. Here you can see our multimodal microscope platform. This is a custom research setup, and it's developed for nonlinear optical imaging. And besides SRS signal, we can also detect Gauss signal, two-photon fluorescence, and second harmonic generated signals. And this multimodal capability is very important because different mechanisms can provide us complementary information about the same sample. Before moving to tumor imaging, I would like to show you a few examples of how our setups can perform. Well, we have more departments at the company, and one of them was an in-house project where chemists wanted to measure. They had a project which focused on a label-free determination of saturated and unsaturated ratio in esters of fatty acids. Well, these molecules have naturally Raman active functional groups, and this gave the opportunity that they could prepare samples with different but known ratios of saturated and unsaturated fatty acids, and they could determine the ratio also in a voxel size with a very good sensitivity. Another example of what the company has performed, that they also added a lipidophile compound to a embryonic kidney cell line, and after a short incubation time, they could visualize how this tracer compound accumulated inside the cells in intracellular vesicles. Why is this important and why is this interesting? Because these tracer compounds added to tissues can help to understand how drug delivery works. I forgot to mention that this experiment was done in the cell silent region, and the first one was in the fingerprint region, sorry. Okay, and our setup is also capable of measuring one after each other at different wave numbers. Here you can see, as an example, a fixed deparaffinized mouse pancreas, and by collecting all the images at the different wave numbers, you can choose a spot which you can tell what kind of subcellular part of the cell is, and we can also plot its spectral behavior. We have a collaboration with the pathologists from the Semmelweis University, and we chose to investigate the the pancreatic ductal adenocarcinoma. And why this? Because it remains asymptomatic until an advanced stage. And once the first symptoms appear, it progresses rapidly, and it comes with a poor prognosis. The pathologists usually work with hematoxin and iosine stained samples, tissue samples, and in case of pancreatic tumorous tissues, it's sometimes difficult to distinguish from chronic pancreatitis. I have to point out that pathologists do not evaluate a sample cell by cell. They are looking at the sample, at the section as a whole structure, and to evaluate it, they are looking for morphological patterns, spatial arrangements, and microenvironments of the tumorous area. On our setup, we acquire data in a 4x4 mosaic, and all the 16 tiles go into the network, into the artificial intelligence network as an input parameter, one by one, not as a stitched image. Here, I would like to show you some example how our images look like. This is a stitched version, this 4x4 tissue section. This is an example for human chronic pancreatitis, and I would like to point out that these are all label-free images. Here, the second example is taken from a tumorous section. And I also added the autofluorescence of the collagen inside the tissue. And this is the anti-Stokes or Cars signal of the sample. And at the different wave numbers, here you can see nuclei, and sometimes these wave numbers are chosen where the lipids and the proteins have a higher signal. Okay, at the right-hand side, if you are not familiar, I would like to show you how a hematoxylin eosin stained sample looks like. It has an excellent morphological contrast, and sometimes it is possible to generate hematoxylin eosin-like image from raw SRS data. But here I have to tell you that this project started at the company during the COVID era, which meant it was really impossible to get fresh or frozen samples, but the project had to go on. So we decided to go on with this fixed deparaffinized samples. That's why we had to measure these samples at uncommon wave numbers. What happens from here? Well, the hematoxylin eosin-like image is not possible to generate from these images, but the question remains still open whether we can teach or learn the structures, some morphological structures related to tumorous patterns with the raw SRS data. All the tissue regions, all the 16 tiles were annotated by pathologists one by one. These labels included tumorous tissue and many different components which can be found inside the human pancreas. We used supervised deep learning with deep learning, meaning that the model was trained on images where the labels were also known for the system. And that's why the goal was to learn the relationship between SRS image patterns and pathological tissue categories. For classification, we use the residual convolutional neural network, or ResNets, with many different layers. So we tried it with different layer depths. And in our workflow, the ResNet acts as a feature extractor. So it learns the image features from the SRS tiles, from simple texture to more complex tissue-related patterns. Then these features are passed to a fully connected classification subnetwork, which outputs the class probabilities. The first task was binary classification. This means that the network had to decide which tile is tumorous or non-tumorous. Well, this is the most clinically relevant first question. And our results show that the SRS image data contain enough information to learn tumor-related patterns. And this supports our central message here that even when direct stimulated Raman histology like images cannot be done, then AI can still extract relevant information for diagnostic. And the next step was multi-class classification, where the model was trained to distinguish between the several tissue categories I have showed you previously. This is a more difficult task because in a real-life tissue, so a real-life tissue is heterogeneous, And this means that a single tile may contain mixed components. Nevertheless, multi-class classification is still important because it moves the analysis closer to real histopathological interpretation, where the tissue context also matters. So although at the moment binary and multi-class classification are available only by using raw SRS images, but multi-label analysis is still under investigation and development. Thank you for your attention.
Thank you, Vicky. Anyone has a question? Okay.